Welcome to my course on microbial biotechnology. Today we are going to start module 2, which will deal with the structure and life cycle of representative groups of virus, prokaryotic and eukaryotic microorganisms. Let us start with the structure, life cycle and classification of virus, because viruses are altogether different from the other microorganisms. So, this lecture is divided into 3 sections. We will start with an introduction to viruses, where we will be discussing about the general structure of viruses regarding their genome size, viral particle size, viral proteins and morphology. Then we will go on to discuss about the life cycle of viruses and one-step growth curve. Then finally, we will deal with the virus classification. We will have introduction to various virus classification schemes, and then deal in length with Baltimore classification. So, let us try to understand what a virus is. A virus differs from living organisms as they lack organelles, cytoplasm or a nucleus. They consist of 2 main components, a nucleic acid core and a protein coat. What is this nucleic acid core? A virion contains either DNA or RNA, but not both at the same time, and these can be single stranded or double stranded, linear or circular. Some viruses like influenza have segmented genomes. Now, what is the protein coat? The protein coat, basically referred to as the capsid, is composed of protein subunits called capsomers, and these gives virus its shape and symmetry. In this picture, you can see the structure of a coronavirus with some very interesting features on its surface. We will discuss about these in length today. The capsid and enclosed genome together form the nucleocapsid, which protects the nucleic acid against environmental changes, but there are cases where virions do not contain any capsid and the nucleic acid stays naked. Many viruses have spike proteins, as you can see in the case of coronavirus in the earlier picture, on the capsid to help attach to and penetrate host cells. As I already told you, there are certain naked viruses, they have only nucleocapsid without an envelope. Envelope viruses have an additional lipid and protein membrane surrounding the nucleocapsid. They also contain a matrix layer between the capsid and envelope. In brief, viruses are obligate intracellular parasites. They cannot replicate without a host, making them intermediates between biotic and abiotic forms, or what we in general call as the living and the dead. They infect organisms of all 3 domains: eukarya, bacteria and archaea. Since the discovery of the first virus, tobacco mosaic virus, in the mid 1800s, over 11,000 virus species have been detailed. However, this represents only a small fraction or minuscule, as millions of virus species most likely remain undiscovered. So, this is the atomic model of a SARS-CoV-2 virus in A, which we have already shown you in earlier slide, and in B you can see a tomato leaf infected with tomato TMV, tomato mosaic virus, and number C, a tomato leaf infected with TMV, another picture, and in D you can see the atomic model of T4 bacteriophages. What are bacteriophages? We will be discussing soon. So, viruses are often described as organisms at the age of life. We refer to it as in between the biotic and abiotic forms, and therefore, they exhibit both biotic and abiotic characteristics. What are the biotic and abiotic characteristics exhibited by virus? Let us start with the biotic properties of viruses. We already told you that they contain either DNA or RNA as the genomic material, like other living organisms who have nucleic acid, mostly DNA. Virus can evolve by natural selection, generally through mutation. In fact, the mutation rate in virus is very, very fast. That is why influenza virus, which mutates very fast, or several other viruses which mutate very fast, are challenges to therapeutic interventions, because we cannot develop stable vaccines against them. Viruses can reproduce by creating multiple copies of themselves, but abiotic forms or components cannot do this. So, in this way they are similar to the living world. What are the abiotic properties of viruses? So, the viruses lack a general cellular organization with a living membrane and protoplasm. They lack general translation machinery and therefore rely on host cells for reproduction. Viruses undergo self-assembly, unlike life forms which reproduce through cell division. Viruses can be crystallized as they are proteinaceous. So, these are some of the abiotic properties of viruses. Now, if you look into the general structure of viruses, we have already described that it can be described as a nucleocapsid molecule with a single nucleic acid molecule surrounded by a protein coat, which we know call as the capsid. This capsid is made up of structural units called capsomers, together held by non-covalent bonds, and plays a crucial role in virus morphology. Some complex viruses have capsids enclosed within a lipoprotein envelope derived from the host cell membrane. Each virus has a single type of nucleic acid in its genome, ranging from 7,000 to 20,000 base pairs, which can be either DNA or RNA. Viral nucleic acids can be double stranded or single stranded. The Hypantiviridae family is an exception with genomes that are partially double stranded and partially single stranded. Additionally, single stranded RNA viruses are classified as positive strand or negative strand based on the complementary to viral mRNA. Now, how big or small a virus particle is? Generally, a virus size could range from 30 to 400 nanometer, making them 100 to 1000 times smaller than the cells they infect. Parvovirus and adeno-associated viruses have diameters around 20 to 30 nanometer. There are some giant viruses which typically range from around 200 to 400 nanometer in size and have genome sizes around 1.5 million base pairs. Filoviruses like Ebola and Marburg have diameters of about 80 nanometer, but can be over 1000 nanometer in length due to their thread-like structure. Pandoviruses are the largest known viruses, measuring around 1000 nanometer in length and with genomes of about 2.5 million base pairs, exceeding some bacterial genomes and comparable to those of certain single-celled parasitic eukaryotes. So, here you can see the TAM image of Pandora virus, and this is something around 500 nanometer scale, and you can very well make an assessment of the giantness of this particular virus. Now, let us discuss about the viral proteins. Virus code two type of proteins. The number one type is the structural proteins and the second type is the non-structural proteins. So, these structural proteins form the capsomers that create the capsid, providing a protective coating for the viral nucleic acid. Each capsomer typically consists of 1 to 6 polypeptide molecules, which can be homopolymers or heteropolymers. The non-structural proteins coded by virus are essential for viral replication processes. Some of the examples of these proteins are transcriptases and polymerases, which are directly involved in replication of viral particles. Then there are regulatory proteins which control the expression of genes. Then there are proteins involved in packaging of genome and assembly of viruses, and proteins involved in perturbation of immune system. How does a virus look like, or what is the physical appearance of a virus or virus morphology? They may be helical in appearance. These viruses are constructed with capsomers forming a helical virus around a central axis. Variants can take form of short and rigid rods or long and flexible filaments, and in this electron micrograph of tobacco mosaic virus you can see these particular shapes. Then there are viruses which are icosahedral. These viruses exhibit a symmetrical, roughly spherical shape with 20 identical triangular faces. Capsomeres are of two types: pentons, which form vertices, and hexons, which form faces. And here you can see the electron micrograph of an adenovirus. Then there are certain viruses which are called as head and tail viruses. Bacteriophages have a structure with a elongated icosahedral head and a tubular tail. The tail serves as a delivery mechanism for the viral genome. Then another type of morphology is pleomorphic viruses. These exhibit varying shapes or forms, as you can see in this picture. The overall shape of pox viruses can vary from ovoid to brick shaped. Then we have envelope viruses. These are viruses which contain a viral envelope obtained from host cell membrane. This envelope consists of a lipid bilayer with proteins encoded by both viral and host genomes. In the picture you can see the electron micrograph of SARS-CoV-2 virus. Let us now move on to the second section of this lecture, which deals with the life cycle of viruses. The life cycle of viruses can be easily explained with one-step growth curve, which is introduced by Max Delbruck and Amory Ellis in 1839. The viral one-step growth curve describes the stages of viral replication within host cells, including attachment, entry, eclipse, replication, assembly, and release. Unlike the sigmoidal bacterial growth curve, it consists of these following phases. The first phase is the inoculation, where the virus bind to the host cell. The second step is the eclipse, where particles penetrate the host cell. The third step is the burst, where the host cells release virions, and the fourth is the plateau, where virus particles are released. So, these diagrams shows a detailed view of the viral one-step growth curve with the latent phase and the eclipse phase and the burst size. Let us look into the various phases one by one. The inoculation phase. When a host is inoculated with a virus, the initial viral count is high, but gradually decreases as viruses attach to host cells via specific receptors. This decline occurs due to saturation and can also be manipulated by adding antibodies to the media. The second step is the eclipse phase. After attachment, viruses enter the host cell, where biosynthesis of components for new variants occurs. In the later stage, virion assembly begins, but the total number of viruses inside and outside the cell remains unchanged during this phase. In the latent phase, here we consider the period from attachment until new viral particles bud or emerge from the host cell. Mature variants will accumulate inside the cell, increasing the viral load, but no extracellular particles appear until the threshold is reached for budding. The next phase is the burst phase. Once the retention threshold is reached, cells either lyse, that is break up, or release virions by budding, causing a sharp rise in extracellular virions, and you can see the virus particles coming out here. Eventually, no more viruses are released, and we can estimate the number of virions released per infected cell, called as the burst size. Overall, this is the life cycle of viruses within a host. The first is the attachment. The virus is getting attached through a receptor here, as you can see. Then the next step is the penetration. The virus will penetrate the host cell and it will put the viral particle inside the host cell. Here the uncoating will happen, so the nucleic acid is released or the viral genome is released, and then these viral genome will undergo various molecular events like transcription, translation or replication. So, this is the step of biosynthesis, and where both the viral genome is copied as well as the viral proteins are produced. These two together will then go for self assembly, as you can see here, and the viral core structures are formed and finally released, and we obtain the mature virions. So, from attachment to penetration to uncoating to biosynthesis to assembly to release, we consider as a life cycle of a virus overall. So, a little bit of detail regarding these various stages of the life cycle. First, the attachment process involves the initial interaction between virus particles and the host cells. It includes two main components: viral receptors located on the outer surface of the host cell membrane, as we have already discussed or mentioned in the earlier slide. Then viral anti-receptors found on the viral capsid and or envelope. Certain co-receptors may also facilitate the interaction between viruses and their receptors. It is important to note that viral receptors serve essential cellular functions beyond just binding to viruses. The presence of these receptors is crucial in determining whether a specific cell type is susceptible to infection by certain viruses. If the receptor is absent, the cell will not allow the virus to attach, and therefore it will become immune to it. Consequently, receptors play a vital role in cell tropism, which refers to the ability of viruses to infect specific cell types. So, here is a summary of the viral receptors and tropism for major human viruses. We have for ease of discussion divided these viruses into DNA viruses and RNA viruses and reverse transcriptase viruses or retroviruses. So, there are various examples like parvovirus, polyoma, adenovirus, herpes virus, and then picornavirus, then flavivirus, then coronavirus, and then orthomyxovirus, rhabdovirus, which causes rabies, and then we have retrovirus, which causes HIV and hepatitis B virus, and these are very, very challenging problems still haunting the world. So, you can see the corresponding receptors in this column. For example, parvovirus, the receptor is HSPG, and then for polyoma it is GM1 and so on. For coronavirus, you have this ACE2 receptor, and you have the CD4 for HIV1 and anti-CP for the hepatitis B virus. And then there are certain viral anti-receptors. For example, you have these CAP in the case of parvovirus, and then BP1 in the case of polyoma, and so on. And in the case of coronavirus, you have this spike, and then you have these GP120 in the case of HIV, and pre-S1 in the case of hepatitis B virus. So, based on these tropism, they can be classified as, for example, variable neurotropic, or they may be variable oral epithelium, beta cell and epithelial cells. Then they may be hepatotrophic, or they may be having tropism towards the respiratory tract, Okay. And then they may be having tropism for the macrophages and so on. Next comes the penetration. The penetration mechanism varies by virus types. Envelope viruses primarily enter cells via two mechanisms. Number one is the direct fusion and receptor mediated endocytosis. In contrast, non-envelope viruses mainly use receptor mediated endocytosis for penetration. Direct fusion. Direct fusion is a mechanism where the viral envelope merges with the cell membrane, allowing the viral nucleocapsid to be delivered directly into the cytoplasm, while the viral envelope remains on the cell's plasma membrane. So, here you can see the mechanism of direct fusion, where there is a host cell receptor and the fusion of the host cell membrane and viral envelope taking place soon after these attachment through the receptor, and the virus is thereby internalized subsequently. So, retroviruses such as HIV use this mechanism to penetrate the host cells. Receptor mediated endocytosis. This involves receptor ligand interactions that trigger endocytosis, leading to the formation of a coated pit and subsequent endosome formation enclosing the virus particles. So, you can see here the formation of the coated pit. Envelope viruses, the endosomes break down via fusion of the viral envelope with the endosomal membrane triggered by acidic pH. In the case of non-enveloped viruses, the induced endosome lysis through one of their capsid proteins. Typically, receptor mediated endocytosis follows a clathrin dependent pathway in this case. The third step is the uncoating during viral replication. As virus particles move from the cell's periphery to the perinuclear space, or they are internalized, the viral genome is exposed to the cellular machinery for gene expression. This process, known as uncoating, is often associated with the endocytic pathway or cytoplasmic trafficking. For viruses that replicate into the nucleus, the viral genome must enter through nuclear pores. Similar viruses like polyomaviruses allow the viral capsid to enter the nucleus. In contrast, larger viruses dock their nucleocapsids to nuclear pore complexes, leading to partial capsid disruption that enables the DNA genome to enter the nucleus. In the biosynthesis stage, viral genome replication strategies vary significantly among virus families. Distinguishing each family, we will discuss these soon in this lecture. The extent to which virus families rely on their host cellular machinery also varies, from complete dependence to relative independence or partial independence. The same is true for viral replication. However, all viruses rely entirely on the host translation machinery, whether complete or relative, specifically ribosomes, to synthesize their proteins. In the assembly and maturation stage, capsid assembly occurs. As the viral genome and proteins accumulate within the infected cells and involve two processes, number one, the capsid assembly and genome packaging. These procedures may happen sequentially or simultaneously. For example, picornaviruses exhibit sequential assembly, while adenoviruses demonstrate simultaneous assembly. In picornaviruses, capsids or procapsids are assembled without the RNA genome, which is later packaged into the procapsid through a pore. So, the capsid will be procapsid will be formed, and through this pore the genetic material will be internalized. In contrast, adenoviruses couple capsid assembly with DNA genome packaging. So, it goes side by side, where viral capsid proteins recognize a packaging signal within the genome, allowing for selective packaging of RNA or DNA. Finally comes the release phase, where the viruses are released. In the case of naked viruses, virus particles are released through cell lysis, where infected cells rupture. disrupting the cell membrane that contains the assembled viruses. Therefore, no specific exit mechanism is required in the case of naked viruses. In the case of enveloped viruses, they undergo envelopment where viral capsids are enclosed by a lipid bilayer before release. If you compare these to the case of sending letters in older times, here you write a letter and then put the letter inside an envelope and then seal it. But in the case of naked viruses, you do not use any paper, you directly use a postcard to write your message and send it. So, these are the two different release mechanisms. So, these can occur after case of capsid assembly with fully assembled capsids recruited to the membrane through interactions with viral envelope glycoproteins. Alternatively, envelopment may occur simultaneously with capsid assembly. Let us now discuss about two important topics in virus life cycle, which is lysis and lysogeny. The life cycle of bacteriophages serves as a valuable model for understanding how viruses impact infected cells. Similar processes are observed in eukaryotic viruses, which can cause immediate cell death or establish latent or chronic infections. Virulent phases typically lead to cell death through lysis, while temperate phases can integrate into the host chromosome, entering a latent phase known as lysogeny. During this phase, they replicate along with the host genome until they are induced to produce new progeny viruses. So, here we can see the two different life cycles, the lytic cycle on the top and the lysogenic cycle below, and also how these two life cycles are interconnected. So, here we can see that after the attachment and internalization of the virus into the host cell, a multiplication of proteins and DNA is happening, and they are getting self assembled into virion particles, and the cell is broken open here to release the mature virus particles. So, this is known as lysis, which kills the cell, host cell. In the case of lysogeny, once the viral genome is internalized, the viral genome gets integrated with the host genome. So, it becomes a part of the host genome. Now, when the host genome will replicate, as you can see here, the integrated viral genome will also get integrated. So, from one copy it become two copy here along with the two host daughter molecules, and then as the cell divides to distribute the genetic material, both the daughter cells will have a copy each of the viral genome along with its own host genome. So, this cycle can go on for quite a long time, but then finally the virus will undergo certain molecular events due to which these integrated viral genome at a particular point of time will get released through various mechanisms and also produce the proteins which is required for its packaging, and then it will enter the lytic cycle. So, a virus which was in the lysogenic cycle, in order to be released as mature viruses, has to undergo a transformation in its life cycle. Not necessarily it goes into the lytic cycle, it can be also released without lysing the host cell by other mechanisms. So, during the lytic cycle of a virulent phase, the bacteriophage takes control of the host cell, replicates new phases and ultimately destroys the cell, which we have already discussed in the earlier diagram. These cycle consist of 5 distinct phases which we have discussed at length earlier, which includes the attachment phase, then the penetration phase, and then biosynthesis, and then finally assembly and release. So, overall, starting from the attachment, we go for the penetration phase, and then the biosynthesis phase, and the assembly, and then finally release. In the lysogenic cycle, the phase genome enters the host cell through attachment and penetration, but integrates into the bacterial chromosome, forming a prophase instead of killing the host. The infected bacterium is called a lysogen, and the process is known as lysogeny. Temperate phases typically remain latent within the cell. As the bacterium replicates its chromosome, it also replicates the phases DNA, passing it to daughter cells during reproduction, which we have discussed with the help of the diagram earlier. The prophase persists in the host chromosome until induction, which removes the viral genome, allowing the temperate phase to proceed to the lytic cycle. So, here is the lysogenic cycle, which was already discussed, starting from the attachment, and then the internalization of the DNA, and then formation of the prophase and distribution, replication and distribution into the two daughter cells, and then already discussed at some point viral DNA is excised from chromosome and becomes active. Then the host replicates viral DNA, builds viral proteins, and then formation of new virus, and then goes on to the cell lysis and release of new viruses. An important concept is the lysogenic conversion. The presence of a phase can alter a bacterium's phenotype by introducing additional genes such as toxin genes that increase virulence. This change is known as lysogenic conversion or phase conversion. Without the prophase, bacteria like Vibrio cholera and Clostridium botulinum are less harmful. When infected by phases carrying toxin genes, these bacteria becomes more virulent. For example, the phase encoded toxin in Vibrio cholera can cause severe diarrhea, while in Clostridium botulinum it can lead to paralysis. So, here you can see the lysogenic conversion expression during lysogeny. So, this is a phase encoded toxin which is coming from the viral genome. So, this is the bacterial genome into which this viral genome has got integrated along with the toxin gene. So, here is the case of lysogenic conversion, where the lytic subpopulation phenomena can be observed, where is the prophase with toxin, and then here it produces phase encoded toxin, and induction of the lytic and toxin genes which has got integrated here results in this phase encoded toxin, which leads to the lysis. And then here you can see the host cell invasion. Let us have a comparison between lysis and lysogeny. In the phase genome, three categories of genes influence whether a lytic or lysogenic cycle would occur. Number one is the immediate early genes. These genes are activated by host RNA polymerases and include CRO, CII and N. CII acts as a transcription factor, stimulating the expression of the primary lysogenic refresher gene CI. CRO represses the CI gene expression. The competition between CRO and CII determines the production of CI gene repressor. If CII activates CI expression, sufficient CI represses early promoters, shifting the infection towards lysogeny. N functions as an anti-termination factor necessary for transcribing the delayed early genes. The delayed early genes includes replication genes O, P and Q, which encodes the anti-terminator necessary for transcribing late genes. Q is crucial for continuing transcription and expression of these genes. Late genes in lambda phage, 5 proteins are involved in lysis: choline and anticholine from the S gene, endolysin from R gene, and spannin proteins from genes RZ and RZ1. The timing of lysis is regulated by choline and anticholine, with anticholine inhibiting choline. In T4 like phases, lysis can be inhibited by genes R1 and R3, which block the T4 choline during superinfection by another T4 or closely related variant. Let us now discuss about the classification of viruses. So, in this section we will be discussing at length about the Baltimore classification of viruses. Before that, let us look into other kinds of classification, particularly based on nucleocapsid symmetry. So, here you can see a simple classification of viruses which are based on their nucleocapsid symmetry. For example, viruses can be described as helical, like rabies and tobacco mosaic. They have a coil shape resembling a corkscrew, as you can see over here. Then viruses can be having icosahedral symmetry. These viruses such as herpes simplex and poliovirus have 20 triangular faces and 12 corners, and you can see the beautiful geometric shape over here. Then we may have certain complex symmetry like these in D. So, these includes viruses like bacteriophages, which have an icosahedral head, you can see on the top, and a helical tail seat. Then we may also have brick sepate viruses. For example, pox virus is a brick sepate with filaments arranged in a swirling pattern around the periphery. Virus classification based on the ICTV system, or the International Committee on Taxonomy of Viruses, which was established in 1966. So, this is less precise than that of cellular microorganisms due to limited knowledge of their organisms and evolution. These ICTV provided a uniform classification system based on nucleic acid types, envelope presence, capsid symmetry, and variant dimensions. The taxonomic levels include here order, virals, for example, codovirals, then family Viridae, example herpes viridi, then subfamily Virini, example herpes virini, then genus otovirus, example herpes virus, then species descriptive names like human herpes virus 1. The ICTV system is continuously updated to reflect new discoveries and ensure global consistency in virus taxonomy. Let us have a detailed look of the Baltimore classification. So, this was developed by virologist David Baltimore in 1970s, and it categorizes viruses based on 4 main criteria. Number 1 is the type of nucleic acid, then strandedness, then the sense, and the method of replication. When we tell type of nucleic acid, what type of nucleic acid this particular virus has, DNA or RNA. When we tell strandedness, whether it is single stranded or double stranded. And for single stranded RNA viruses, either positive sense plus or negative sense minus must be converted to positive sense rRNA in this case. And in the method of replication, how the virus replicates its genome and produces mRNA in the host cell are the important criteria in this classification. The Baltimore classification organizes viruses by these characteristics, allowing for the study of similar behaviors within distinct groups. Initially, it had 6 groups, which were later expanded to 7. We will look into these 7 Baltimore classes or BC. So, these are divided into group 1 to group 7. For example, you have group 1 double stranded DNA viruses. in this case. Okay. Then you have group 1, which is single stranded DNA viruses. Then group 3, double stranded RNA viruses. Then group 4, positive sense single stranded RNA viruses, and then group 5, which is negative sense RNA viruses. Then group 6 is single stranded RNA viruses with a DNA intermediate in their life cycle. Then group 7, double stranded DNA viruses with an RNA intermediate in their life cycle. So, these particular diagram comprehensively describes the 7 groups in the Baltimore classification. So, we have seen the 7 groups based on the nucleic acid they include, or the strandedness, and the intermediaries in their life cycle, RNA or DNA in between. And if you look into the mode of mRNA production from 1 to 7, in the first case mRNA is transcribed directly from the DNA, for example in herpes simplex virus. In group 2, like those in canine parvoviruses, DNA is converted to double stranded form before RNA is transcribed. In group 3, mRNA is transcribed from the RNA genome, for example in the case of rotavirus. Then in the fourth case, the genome functions as the mRNA because this has a single stranded positive RNA. Picornaviruses, which causes common cold, are classic examples of these. Then in the case of single stranded negative RNA, mRNA is transcribed from the RNA genome, such as in the case of rabies. Then in sixth group, reverse transcriptases make DNA from the RNA. DNA is then incorporated in the host genome. mRNA is transcribed from the incorporated DNA. So, the best example of these is the human immunodeficiency viruses. In the seventh group, the viral genome is double stranded DNA, but viral DNA is replicated through an RNA intermediate. The RNA may serve directly as mRNA or as a template to make mRNA, for example the hepatitis B virus. So, let us look into a little bit of details of the class 1 viruses. The class 1 includes viruses with double stranded DNA, as already mentioned. Like cells, these viruses transcribe their DNA asymmetrically to produce messenger RNA. Some double stranded DNA viruses replicate in the nucleus relying on the host polymerases, while others replicate in the cytoplasm using their own mechanisms for transcription and replication. Examples include herpes viridae 1, 2 and EBV, which causes chickenpox, singles and infectious mononucleosis. Then we have papilloma viridae, these are responsible for warts and cervical cancer. Then pox viridae, this causes smallpox, monkeypox and vaccinia viruses. In this picture we can see the electron micrograph image of an herpes simplex virus on the top, and in the bottom we can see the smallpox virus. Let us now look into the replication and translation of Baltimore class 1 type of viruses. The replication of double stranded DNA viruses involve several mechanisms. The first one is the bidirectional replication, where two replication forks move in opposite direction from an origin, but this model has now been revised where in fact the movement is in the same direction due to the involvement of one single polymerase, and there is a looping which occurs to accommodate the steric hindrances resulting out of these kind of complex structures. We will not discuss about that in length. Those who are interested can refer to the replication fork model, revised replication fork model. The second one is the rolling circle mechanism, where linear strands are produced as a circular genome loops around. And the third one is the strand displacement method. One strand is synthesized from a template followed by the synthesis of a complementary strand, resulting in double stranded genome. The fourth one is the replicative transposition. The viral genome replicates within the host's DNA and relocates to another part of the host genome. mRNA synthesis occurs in 3 steps. A transcription pre-initiation complex binds to the DNA upstream of the start site, recruiting the host RNA polymerases. The RNA polymerase uses the negative strand as a template to generate mRNA. The transcription ends when RNA polymerase reaches a specific signal such as a polyadenylation site. In the class 2 viruses, which are single stranded DNA viruses, they include a single stranded DNA genome. These viruses also undergo asymmetrical transcription to produce messenger RNA. However, upon entering a host cell, the single stranded genome is first converted into a double stranded form from which mRNA is synthesized. Some of the examples include parvoviride, which causes illnesses like Fifth disease, and then inoviride includes bacteriophages that infect E. coli and Pseudomonas, then Geminiviride, plant viruses that causes disease like leaf curling and yellowing. So, here is the electron micrograph of canine parvovirus on top, and then we have these on the bottom, the Gemini virus, and you can see these particles always occurring in pairs, as the name Gemini suggests. Replication and translation in these Baltimore class 2 type of viruses. Eukaryotic single stranded DNA viruses primarily replicate in the nucleus. Most have circular genomes that utilize rolling circle replication. In contrast, parvoviruses have linear single stranded DNA genomes that replicate via rolling hairpin replication, which is similar to rolling circle replication. Parvoviruses can package either the positive or negative sense strand into capsids depending on the virus. The newly formed single stranded DNA may be packaged into variants or replicated by a DNA polymerase to create a double stranded form for transcription or further replication. The class 3 double stranded RNA viruses. Since double stranded RNA is not naturally found in cells, it can trigger detection and neutralization by cellular mechanisms. To avoid this, many double stranded rRNA genomes are encapsulated within capsins, which protects them from host detection, and it is a very clever way by the virus to save itself. So, some of the examples include Rioviride, rotavirus, which causes gastroenteritis in young children, and orthorioviruses, they link to respiratory and gastrointestinal illnesses. Then Birnaviride, infectious bursal disease virus. Then Totiviride, for example, Saccharomyces cerevisiae are some of the examples. Here you can see the TAM image of piscine reovirus on the top, and then we can see the TAM image of rotavirus below. How does replication and translation happen in Baltimore class 3 type of viruses? Upon entering a host cell, the double stranded RNA genome is transcribed by the viral RNA dependent RNA polymerase to produce mRNA from the negative strand. This mRNA can then be used for translation or replication. The mRNA exits the capsid for translation in the host cytoplasm to produce necessary proteins. For replication, the mRNA is transferred from a measured capsid to a progeny capsid. The viral RDAP replicates single stranded mRNA to generate the double stranded RNA genome. The 5' end of the double stranded RNA genome can be in various forms such as naked, capped, or covalently bound to a viral protein. Let us now look into the BC4 type of viruses, which are positive sense single stranded RNA viruses. They function directly as messenger RNA, eliminating the need for transcription prior to translation. During genome replication, intermediate double stranded RNA molecules are produced, which can trigger the host's immune responses. To avoid detection, these viruses replicate within the membrane associated vesicles that serve as replication factories. So, some of the examples include flaviviridae, which causes for example, dengue, then Zika and hepatitis C, then picornaviridae, enteroviruses, and then rhinoviruses, hepatitis A virus, then coronaviridae, which caused the recent pandemic, SARS-CoV-2. So, here you can see the TAM image of Zika virus on the top, and then we can see the TAM image of polioviruses on the bottom. How does replication and translation happen in Baltimore class 4 type of viruses? So, these positive single stranded RNA viruses create positive sense copies of their genome from negative sense strands of an intermediate double stranded RNA, functioning as both transcription and replication. The 5' end can be naked, capped or protein bound, while the 3' end may be naked or polyadenylated. These viruses often selectively transcribe portions of their genome, producing subgenomic RNA for translating structural and movement proteins. As the RNA transcription can initiate within the genome, terminate at specific sequences or attach sequences from viral RNA. RNA dependent RNA polymerases RDRP is always translated first, as it is essential for subgenomic RNA synthesis. Next come the BC class 4. These includes viruses with negative sense single stranded rRNA genome. In these viruses, positive sense mRNA is transcribed directly from the negative sense genome. Some negative single stranded rRNA viruses are ambisense with both strands encoding viral proteins and producing separate mRNA strands from the genome and its complementary strand. Some of the examples include filoviridae, the Ebola virus, then measles virus, mumps virus, and then we have influenza A, B, C viruses, and then we have the rabies viruses. On top you can see the TAM image of vesiculovirus. Then we see the Ebola virus on the bottom in this illustration by Murphy. How does the replication and translation happen in Baltimore class 5 type of viruses? During transcription, RDRP or RNA dependent RNA polymerase generates a 5' and triphosphate leader RNA. When it encounters a leader sequence, the 5' end capping or cap snatching from host mRNA allows viral mRNA recognition and translation by host ribosomes. RDRP initiates transcription at a gene start signal and terminates at a gene end signal. At the end of transcription, it adds a poly A tail to the mRNA's 3' end consisting of hundreds of adenine nucleotides, often through suturing on uracil sequences. Replication. The genomic negative ssRNA is replicated from the positive sense antigenome similar to transcription, but in the reverse way. RDRP moves from the 3' end to the 5' end of the antigenome, ignoring transcription signals, and uses the antigenome as a template for the genome. Class 6 Baltimore viruses. This includes viruses with a positive sense single stranded rRNA genome that have a DNA intermediate stage in their replication cycle. These positive strand single stranded rRNA reverse transcripted viruses employ a unique strategy involving reverse transcription and integration into the host cell's DNA, allowing them to effectively utilize the host transcription machinery for replication. Some of the examples include retroviruses such as HIV, which causes AIDS, and the human T cell leukemia virus or HTLV. Then we have lentiviruses. These include simian immunodeficiency viruses, which affects primates and is closely related to the human immunodeficiency virus. So, here we can see the HIV virus on the top and HTLV in this TAM image, and then we can see here bovine smoma virus in the bottom panel. The replication and translation in Baltimore class 6 viruses. In this replication of single stranded rRNA RT viruses, their linear RNA genomes are first converted into double stranded DNA through reverse transcription, facilitated by the viral reverse transcriptase enzyme, which synthesizes a complementary DNA strand from the single stranded RNA template. The RNA strand is then degraded, resulting in a double stranded genome. The double stranded genome integrates into the host cell's DNA as a provirus. Inside the nucleus, the host's RNA polymerase II transcribes RNA from the proviral DNA, generating various RNA types including mRNA for protein synthesis and copies of the viral genome for replication. The last class or class 7 includes viruses with a double stranded DNA genome that has an RNA intermediate known as dsDNA RT. In their replication cycle, these viruses have a gap in one of the DNA strands, which is repaired to form a complete double stranded DNA genome before transcription occurs. This class primarily includes the Hepatnaviridae family, which contains the hepatitis B virus. Hepatitis B virus can cause acute or chronic hepatitis, leading to complications like cirrhosis and liver cancer. The only other family of dsDNA RT viruses is Colimaviridae, which includes the cauliflower mosaic virus or CAMV and soybean chlorotic mottle virus. The transcription of double stranded DNA RT viruses resembles that of double stranded DNA viruses, but these viruses use reverse transcription to replicate their circular genome while still in the viral capsid. In the host cell cytoplasm, the cell's RNA polymerase II transcribes RNA strands from the viral genome. Replication of the double stranded DNA genome occurs based on these RNA strands and follows a similar mechanism so that to that in single stranded RNA RT viruses. However, in double stranded DNA RT viruses, replication occurs in a loop around the circular genome. After replication, the double stranded genome can either be packaged into a new viral particles or transported to the nucleus for further transcription. So, with this we come to end of this Baltimore classification. Thank you.